So we're going to need RNA polymerase because we're going from DNA to RNA. So what we're really building is RNA. We need an RNA polymerase. We need it to build us RNA. We also need to know where to start. So we need something in your DNA strand known as a promoter. This tells RNA polymerase where to sit. So it sits down on that promoter and starts to make the mRNA strand from our DNA. So the RNA polymerase attaches to the promoter. It's a stage known as initiation, or the start point. Elongation, it just grows longer. RNA polymerase adds these RNA nucleotides until it gets to what we call the terminator. So the terminator says, stop doing transcription. Stop going DNA to RNA. We're done. We're at the end of the gene. Get lost. Okay? And when we detach from the DNA, we're going to go off then and actually help to form our proteins with the mRNA that was made. So this is kind of what it looks like. RNA polymerase is a fairly large enzyme, surrounds the DNA strand, finds the promoter region, sits down, starts to build an RNA strand, and it will continue to do that until it hits the terminator region. So, like we said, the RNA polymerase is going to sit on the promoter. It's going to go along and read that DNA strand building this purple or pink ribbon that is mRNA. And eventually it'll hit the terminator. When it hits the terminator spot, the RNA polymerase falls off, and the completed mRNA strand is going to leave the nucleus. So RNA polymerase, we said ACE is an enzyme, but it's a protein, and it's going to be inside the nucleus. When we talked about organelles, we said that it's very important to keep in mind that it's not just DNA inside the nucleus, it's DNA and a whole lot of proteins to do this work. So the mRNA itself is going to encode amino acids. So its job is to now go through translation. So in prokaryotes, so our bacteria, the mRNA is going to get immediately converted into proteins. Because remember, there is no nucleus. So this is a much faster process. In eukaryotes, like we said, the mRNA is going to leave the nucleus and go off usually to the rough ER, or the rough endoplasmic reticulum, to be built into a protein. Now the problem is, prokaryotic DNA is very, very simple, right? It's a single chromosome that's a circle. Eukaryotes, we've got 46 chromosomes, tons and tons of genes to deal with, and the problem is we make far more proteins than we have genes. And that should make absolutely no sense to you. If we have a gene that codes a protein, why don't we have as many genes as we have proteins? Well, you need entirely too many proteins. So we have to find out a different way to go about this. So eukaryotic mRNA has things that we call introns and exons. Introns are pieces of DNA, or in this case, pieces of RNA that must be removed before the mRNA even leaves the nucleus. So essentially, this is a cleaning step, or some people call it a grooming step. You're getting the mRNA ready to actually build the protein your body has asked for. The exons are what are going to exit the nucleus and have what we actually want. It has the code for the protein that is needed. This is done through a process known as RNA splicing. So RNA splicing is going to cut out the introns, keep the exons, and allow them to leave the nucleus. The problem is it's still not quite ready. So if we've got exons, so we've got a strand of mRNA, it's not quite ready to go. Because if I send this out, this is the exact sequence that I need for my amino acid chain to make my protein. But remember, the cell is a dangerous place. If I just send out this mRNA, it's most likely going to get attacked by some sort of enzyme that's trying to chop it up and recycle it. So what we actually do is add on a G, a guanine, literally a nucleotide to one end. And on the other end, we add what we call a poly-A tail. Poly-A because it's many adenines, many A's, in a line off of the end of this mRNA. So if an enzyme comes along and goes chomp, 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 takes off these end A's, I don't care. My mRNA is still exactly what I want. The G, it turns out, is added because when we do translation, we need a little bit of space down here at the end to actually get the next step to start. So we'll show you that. So the introns are cut out, the exons are stuck back together. We get a G cap and a poly-A tail. And finally, after those three major things, my mRNA can finally go out and become a protein, or code for a protein. mRNA is not enough. mRNA needs friends. So we know that it's going to use rRNA. Remember, rRNA builds ribosomes. And if we're going to make proteins, we've got to have ribosomes. So we're going to have rRNA. We've got mRNA. And now we've got tRNA, transfer RNA. These are the interpreters. These are the RNA structures. They're actually going to act like enzymes that are going to help us interpret that information from the RNA into amino acids. And the way it does this is if we have AUG at the start of every single mRNA molecule, right? We told you that. So tRNA's job is to come in and bring in with it an amino acid. Well, how does it know if it's carrying the right amino acid? How does it know that that amino acid is supposed to go first? Well, how would it pair up? How do we pair up DNA and RNA? Well, we base pair, right? So A would become T on DNA, right? But it becomes U because we're dealing with RNAs. U replaces a T, right? So T pairs with A. G pairs with C. So AUG is my codon. UAC, which is part of my tRNA molecule, this is something known as an anti-codon. And it will only bring in this amino acid. We told you this was methionine for our AUG. It will only bring in that amino acid if the anticodon and codon pair up. I know this is a lot. Hang on. So this is the tRNA structure. You see this is RNA that is folded. This is folded RNA. So it's going to act like an enzyme. We said it could do this. We've talked about this before. All the pink lines you see are hydrogen bonds. Okay? And at the end, this attachment point is where the amino acid is going to sit. The anticodon is at the other end. And this is how we kind of draw it. Looks sort of like a birthday candle, right? How we draw it in a simplified form. So we said we also need ribosomes. Ribosomes, remember, are rRNA. They're built from that. So again, a folded RNA structure that's going to act like an enzyme. So we're going to put mRNA together with rRNA, also known as a ribosome. and we're going to add in tRNA to actually build this silly amino acid chain that we're trying to get to. Okay, so here you go. This is what it looks like. This is ribosome. Ribosome. You see large subunit, small subunit, big piece, little piece. Okay, tRNA sitting here in the middle, and our mRNA strand being fed through. The simplified structure is the one on the right here for you. The mRNA strand would run here through the middle. Okay? This is what it looks like all put together. We've got the ribosome, the hamburger bun, made of rRNA. We've got our tRNA, our transfer RNA that's carrying amino acids around. And we've got our mRNA that's being read. So to do translation, the process is the same. The phases are the same as in transcription. Finally, something that's repetitive in biology. Initiation, starting point, starts at the cap. Elongation, we grow an amino acid chain. And then we stop with one of our stop codons. Okay? So initiation to start has to actually bring in this ribosome. We have to bring in the ribosome to sit down on the start position, the AUG, and the tRNA has to come in and sit down. Here's all the verbs for you. Here's what it actually looks like. So we've got the small subunit, the mRNA sits down, the tRNA with the anticodon that we already talked about sits down, and it fits, it matches, carrying the methionine. It should. It starts every single protein in your body. So after that is together, then this large subunit comes in to help stabilize everything. That's the job of the ribosome, is to give a place for stabilization for this to occur. Because otherwise, you see, it doesn't do much. It's just holding things in place. But it's a very important step, because without it, we wouldn't get through all this process. So initiation, everything comes together. Elongation, we build this amino acid chain. So the first step is to recognize that codon with an anticodon. Fine. It's part of the tRNA molecule. We said that. Once that sits down, we're going to start making peptide bonds. So we're going to add amino acids. And we're going to do this over and over and over again. Translocation is the moving of the tRNA molecules to bring in amino acids. Finally, once we do this as many times as is needed for the number of codons, we'll get to the stop codon. We know there are 3 of them. We talked about them when we looked at the amino acid table. And when we get to the stop codon, everything just falls apart. The mRNA falls off, the ribosome falls apart, the tRNAs go back out to collecting their amino acids, bringing them in for the next protein. And the protein that you built floats away. Unless this is part of the rough ER, and then it'll be transported in a vesicle to the Golgi for further processing. So we start, everything comes together, sits down, and we're going to keep just moving through, adding more and more and more amino acids until you get to the stop codon. So in eukaryotic cells, transcription is going to be in the nucleus because it's DNA to RNA. DNA is not allowed to leave the nucleus. It has to happen there. And then the RNA goes out into the cytoplasm, attached to the rough ER often, and does translation. We said that translation happens in 4 major steps. We get those amino acids coming in, we build the polypeptide, we keep building and building until we reach the termination spot or the stop codon. So, if you have an mRNA strand and you're going to build a protein that has 100 amino acids in the protein, how many times do you have to do elongation?